Fantastic. Well, thanks to everyone for joining us for another round of our Corgoning Science webinar series. We're very happy this month to welcome back Renee. Uh, Renee had a maternity leave. We just got to meet Layla, her beautiful daughter, who is now 5 months old, but Renee is back to work full time. So she will be the one to reach out to regarding interest. You can reach out to Renee or to me. You probably got an email reminder from her today about this webinar series, so you can always respond to that. So welcome back, Renee. We're really glad to have you, and we thank Jake Kimmel for his pitching in while Renee was out.
Yes, thanks,
So,
Bill.
yeah.
If everyone wants to see, we got our May webinar starting up on the 2nd. If you want to register for that, I have the link here in the chat. We'll have Caitlin King and Laura Lee talking about restoration planning models to support Great Lakes Cisco restoration.
Okay. Thanks for that. And so next month will be that sort of a tag team presentation, which is the way we're doing it this month. And we really appreciate Corbin and Robin for volunteering. A month ago, we did not have anybody for this, but these two have stepped up to give us an April webinar for us. And so I'm going to introduce them both. You'll hear from Corbin first, and then from Robin. Corbin, you can go ahead and get your slides up on deck if you like. Each of their talks is about 20 minutes. Generally, the theme that they're talking about is identifying and characterizing lake whitefish spawning habitat in Lake Erie. So finding new spawning locations. In the case of what Robin will be talking about, and Corbin will be talking about a new project that's underway to describe overwinter conditions on a known spawning reef. So Corbin got his bachelor's and master's degree from West Virginia University. He then got his PhD at Virginia Tech, and he studied catfishes for both his master's and PhDs. He did a postdoc at University of Toledo, working on the St. Clair Detroit River system, and he is currently a research fish biologist at the USGS Great Lakes Science Center, the Lake Erie Biological Station in Huron, Ohio, where he leads the grass carp early life history research program. And we're tickled that he's also doing some of this Corgoning work as well, Corbin. Robin DeBruyne got her bachelor's and master's from Central Michigan University, her PhD from Cornell University. She is also a fish biologist at the USGS Great Lakes Science Center, specializing in fish early life history on the Great Lakes and in their connecting waters. So with that, Corbin looks like he's all set and ready to go. And take it away, Corbin. Thank you.
All right. Thanks, Bill. So just before I get going, just wanted to thank all these folks listed here for their contributions to this work. We have a interdisciplinary team working on this, and one that's missing is Ed Roseman, who retired as we were developing the proposal for this. So thanks to him also for some contributions early on in this. And a special thank you to the Midwest Climate Adaptation Science Center, who's funding this work, as well as USGS for some in-kind support. All right, so my first slide here is going to be trying to sell the viewers of the Corriganine webinar on the importance of Corriganines. So obviously, we're all well aware that whitefish are an important species in the Great Lakes. They provide important commercial fisheries, but are also ingrained in the culture of the region as well, both pre and post colonization. Lake whitefish have recently undergone some population declines. We can see here in Lake Erie some years of high abundance, but also some time periods here where abundance went below some limit reference points. And also in Lake Huron, we see, this is from some work looking at larval fish densities and larval growth, comparing the historical period and the contemporary period in relation to dreissenids. And we can see some declines recently, and a lot of this potentially could be related to changes in recruitment. That study found that larval densities were lower and larval growth rates were also lower following Dreissenid invasions. So looking at some work from Taylor Brown and some co-authors, we can see Lake Whitefish recruitment indexed as relative year class strength here, has quite a bit of variability, and also in many of the Great Lakes has shown some recent declines. And moving forward, so thinking about what some of those potential contributing factors to those recruitment declines, just talked about Dreissenids. Dreissenids could have a number of mechanisms in which they influence recruitment. One of those, which we probably is pretty intuitive, is zooplankton abundance. Fewer food resources obviously isn't great for larval fish. Spawning stock size, one of the central tenets of fisheries management is you have to have enough adults to produce enough eggs to have recruitment. Currents influence larval distribution and dispersal. Climatic factors are also important. Warm and cold. Cold winters are generally thought to be good for recruitment. Warm springs are thought to be good, and these have sometimes influences from these large-scale climatic oscillations, and also there's been some work suggesting that ice cover is also important. And obviously a lot of these kind of work in concert with each other. Cold winters, you're more likely to have ice cover, and so a lot of these are linked, but are also all factors that contribute to whitefish recruitment based on some of the work that's been done previously. So for this particular work, we are interested in ice cover specifically. A lot of the work previously that has found these linkages with ice cover don't necessarily always get into the mechanisms of why ice cover and whitefish recruitment could be linked. There's some hypothesis development that's occurred, but one of the ideas is that ice cover shelters whitefish eggs and larvae from these wind-driven currents that could, one, displace eggs from reefs, or can redistribute sediments, causing the eggs to be buried. And so there's this potential hydrodynamic component to this, that ice cover may influence eggs based on these currents. And so there are other ideas that ice cover changes emergence periods for whitefish, maybe shifting them earlier, also influences on plankton blooms, and also the potential that ice cover could influence light transmission. And so what this boils down to, some work by Zach Amadon reported that most of the years with high age zero lake whitefish capture rates were years with over 90% ice cover. So there is definitely seemingly some important effect there. And so when I've been trying to sort out what the mechanism and what the possible physical habitat mechanisms for whitefish and ice cover relationships, here's kind of where I've gone with some of the work that's out there, and especially the GLFC document developed by Ebner et al. In 2021, looking at these climatic factors, lake scale factors, and reef scale factors. And so, we could spend a lot of time talking about this, and not all the linkages and influences or influencing factors are really spelled out here, but the idea is that ice cover could limit turbulence and currents, which may then change the rates at which eggs are displaced, agitated, or buried with sediments. And so we wanted to look at that, and going back. So that's kind of where this project is going. And just to note, this is kind of a work in progress. We don't have any data, so unfortunately, no data today. But the reason why this is important is that there have been changes in ice cover through time. So this is looking from 1973 to 2019. And there are places in the Great Lakes that are losing ice cover duration at a rate of 2 days per year. In other cases, it's less than a day or there's no trend. But you'll notice a lot of the places that are losing ice are the places where ice exists, like the western basin of Lake Erie and some of these other bays. And it's also important, not only have we lost ice, but it looks like that's anticipated to continue. So these are projections from various climate models. I've noted the delta T here in red. So that would be the anticipated change in temperature from these models. And it shows that ice cover is expected to continue to decline. Depending on the model, it could be as much as 60 days in some places, as you can see here in the eastern basin of Lake Erie. So in order to understand a little more mechanistically how ice cover could influence hydrodynamics on spawning reefs and potentially lake whitefish recruitment, we started this project this winter, looking at water velocity, turbulence, bottom stress, wave characteristics, and ice characteristics in relation to ice cover. And so this is a project where we have instruments in the lake right now collecting these data, and we're excited to see what we find. But the nice thing about this is we would integrate these observations with other existing modeling efforts. And to kind of give you an idea of what we're hypothesizing that we'll see, this is from, I think, the Arctic, if I remember correctly. And so you can see this first panel is ice concentration. We have wind stress and depth. And within this depth panel, the colors indicate velocity. Darker colors, dark red and dark blue, indicate higher velocity. The lighter shades indicate lower velocity. And you can see during these periods of high ice cover, we still have the high wind stress, and you can see this kind of dampening of the effect on velocity. So that's what we're anticipating that we'll see. So here's how our data collection's kind of working. So we have a reef. Boat comes through. We're dropping these moored acoustic Doppler current profilers. Boat leaves. Whitefish come in and spawn. The acoustic Doppler current profiler is collecting data. Fish leave. And we're going to continue doing this. Winter storms come through, still collecting information. We get ice cover. We're still collecting information. And so we should have information throughout the winter on a variety of conditions: ice cover, open water, storms and non-storms. So should have a broad swath of environmental conditions measured. And so here's what our mooring platforms kind of look like to orient you to kind of what's going on here. So kind of the crown jewel, I guess, of our instruments here is the ADCP, which is collecting current velocity, waves, turbulence, and ice. And so it's a 5-beam hydroacoustic device. And each beam kind of has certain things that it's doing. The 4 beams around the outside are collecting the velocity information. The 5th beam in the middle is doing waves and other things. And so that's all powered by a 1900 watt-hour battery, which you can't see. It's actually behind the ADCP there, in an aluminum tube. And so this is, we also have a pop-up buoy acoustic release, like many of you likely work with doing telemetry work. So we'll go and fire that. There's a recovery line that's going to be attached to the buoy. We're also collecting temperature and turbidity information, again, to look at that sediment resuspension component. Temperature and pressure logger, and we've also, to one of the moorings, attached a photosynthetically active radiation sensor to help maybe get some more information on that light transmittance hypothesis that's out there for possibly explaining the ice cover recruitment relationship. Okay, so for site selection, we had a couple parameters that we were trying to work within. One was the proximity to the Lake Erie Biological Station. Obviously, trying to get these things in the water for the wintertime. Weather windows can be kind of short, so we wanted to be relatively close to the office so we could respond to an open window. when we can get our instruments in the water. Depth is important. So the instruments we're using have specific ranges in which they can operate. The ADCPs shown in the mooring that I showed you, its operating range is up to 8 meters. And so we were looking for less than 8 meters of water. We wanted to make sure that we were in a location that regularly gets ice. And so you can see the Western Basin there looks especially attractive. And I think that's ice cover from 2012 illustrated there. We're looking for hard bottoms. One possible risk with this is if we drop the mooring in soft sediments, we may have a hard time getting it back. And we also, not surprisingly, wanted to target areas with known whitefish spawning to make sure that we were collecting valuable data, essentially. And so that pretty much lended itself, and apologies if this is hard to read, I like old maps, going to the Western Basin reef complex. And when we looked a little more closely at this, it seemed pretty obvious that Niagara was the best choice. So for this, we deployed 3 of these monitoring stations, and Niagara gave us kind of the best opportunity to one, get kind of away from, there were some options over here that would have been suitable. We wanted to get away from wind shadows, so we didn't want islands and other areas to kind of block waves and not give us an idea of what would be happening in the in areas with more fetch. So Niagara made a lot of sense based on all the parameters I just mentioned. We were also looking for to collect hydrodynamics around the reefs, but also to put a mooring offshore to get kind of some off-reef measurements for comparison. And so in December of 2024, we deployed 3 moorings. So 2 of these were Nortec Signature 1000s that operate at 1,000 hertz. We also had a Signature 500, which is lower frequency and is better suited for collecting these measurements in deeper water. And so here we are doing a compass calibration to make sure that the device knows which way the waves are coming from. And we deployed these with the research vessel Muskie in 3 different locations, and I'll show you those. So we tried to set 2 of the instruments on kind of an east-west axis across the apex of Niagara Reef. And when we look at more detailed bathymetry, this one looks like it's a little further east of the apex than it does here. And we put the Signature 500 here offshore a little further in deeper water. And so we're excited to see how much the reef itself influences hydrodynamics in relation to ice cover this year. All right, and so as I mentioned, there's no data yet. So this is more of a what information will we get, not actual information at this point. So we're going to get information on the ice cover effects on hydrodynamics. So currents, turbulence, waves, as I mentioned. We're going to get an idea of the influence of the reef on hydrodynamics, sediment resuspension, bottom stress, turbidity, and also from the ADCP, we'll get measures of acoustic backscatter, which we can also use to qualitatively assess sediment loads. We're going to get the influence of ice cover on light transmission. Again, we only have PAR sensors on 1 of those moorings, so we're not going to get any spatial components of that, but we'll have it from 1 of the moorings. And based on conversations with the Lake Erie Committee's Habitat Task Group, they suggested we leave these out longer than maybe we anticipated doing so. So we're going to leave them out into May to get a feel for flows on walleye, eggs and larvae also. So some relevance here for walleye and whitefish development and dispersal. And so I think one of the interesting things that we're going to see that might be relevant elsewhere is there kind of a hydrodynamic shadow on the different sides of the reef? So this is some work from from Jim Markham and co-authors, and they looked at whitefish in relation to this high-density acoustic telemetry array, and found that lake trout, tended to use the east side of the reefs. And so it might be interesting to see what the difference in flows is east to west on Niagara. And so what can we do with this information? We're planning to develop some spawning reef hydrodynamic atlases based on the information we collect, and then also incorporating that with some of the existing lake models. This is going to inform restoration and maintenance projects. So based on what we see with the flows, there may be some opportunities to leverage that to design and restore reefs in a way that maximize the hydrodynamic suitability of these reefs for spawning. It may also inform reef prioritization for those efforts. This work is going to be important for climate adaptation planning, figuring out good ways to ensure that whitefish recruitment can be maximized in a hydrodynamic context under varying climate scenarios that we may encounter in the future. It could also improve understanding of spatial differences in ice and hydrodynamics, ground truth, or even refine existing lake circulation models. Given that these are directly on spawning reefs, there's the potential that these could be used for dispersal modeling with a little more finer scale than maybe we can do in a lot of cases. Also inform stocking locations and inform experiments. And that was kind of one of the initial reasons we were doing this. We wanted to look at some lab studies with mock reefs in the lab and see what flow levels would eggs be displaced from our mock reefs. And so this is going to be the first step. And so we think getting some of these measurements on the reefs might be very informative for studies within flumes. And so as we kind of move toward the end of this presentation, one thing that we were incredibly excited about was this year being a very good ice year. That was kind of something we weren't really sure about the whole time, was would we get enough ice for meaningful measurements? And luckily, Lake Erie got over 90% ice cover this year. So we're excited to see how things differ in the iced over and ice-free periods. Yeah. So I think that's pretty much it for me. I appreciate everybody tuning in, and I'll be glad to answer any questions at the end.
Fantastic. Thank you, Corbin. We are going to go on to Robin next, and then I should have said that earlier, we'll do questions for both Corbin and Robin at the end of Robin's talk. But you can also put your questions in the chat if you want, if you are worried about forgetting them by the time by the time it comes. So thanks, Robin.
Yep. So good afternoon, and thanks everyone for coming. So I'm going to give a talk that Justin actually gave at IAGLR. So some of you may have seen most of this content before. We did some experimental sampling, monitoring in the central and the eastern basin to try to identify some areas where Lake Whitefish were spawning. I want to start by really thanking all the co-authors here and the field crews that went out for 2 different years in the winter. We had 3 separate field crews each year. Was the funding source, which was the Great Lakes Restoration Initiative Crigony template. So reasons for the study. Corbin went over a whole lot of these, so I'll be real quick with this. But the biggest thing is you can see right here this is for Lake Erie, the recruitment really went down around 2013 to 2015. And that's when our lab in University of Toledo started doing some early life history studies in the Western Basin specifically. And part of those studies was one, figuring out and really compiling where those historical spawning sites were that we could find. That was work led by Hannah Schaefer, as well as from Zach Amadon, who went out in the boat and sampled many locations in the Western Basin just for presence and absence of eggs to see where they were spawning right now compared to where they were spawning before. He also did a whole lot of larval sampling really throughout the whole southern half of the Western Basin. And in 2017 and 2019, this map is only showing results of the 2017 data, but in both of those years, we did larval surveys all down the southern coast for Crigonines, Lake Whitefish, and we caught them. We caught them at all of the ports that we sampled, which they're labeled here. And in 2019, we also caught them here in Cleveland as well. So we know that we're getting larvae throughout the southern shore, but we weren't sure, are they spawning in these local areas, or are these larvae that are, drifting from the west and going down the shore that way? Additionally, the Crigonine framework, restoration framework, has these different stages, which is a theme, obviously, for all of these talks. But this work fits into the gap analysis section and the data, the compiled data to look at the population viability. So if we're looking at whitefish in Lake Erie, we need to know where they're at, where they're spawning, is that successful, and how many are out there. So the study objectives that I'll cover today is one, to verify the contemporary spawning habitat in the central and eastern basins. I mentioned Zach did a phenomenal job really looking at the western basin. And again, this was guided by those historic spawning locations, model results, as well as those larval collections. And in the summer, so we did all of our egg sampling, obviously, in the winter. And then during the summer, they went out and we did some habitat sampling to characterize the substrates of where of the sites we sampled, whether or not we captured eggs. So for site determination, this is sort of just the process we went to. We went from one to the next. So we started with those historic spawning locations. And then from there, one thing we had to look at was, where can we even get in the water along Lake Erie? So then we had to look at which of these locations had access points. From each port, we put a grid up of about 6 miles around. That's what we figured we could travel in a day. If we had, good weather, we could get from one end to the other. Our main sampling strategy was using an egg pump, and that has a max depth of about 8 meters to be effective. So then we limited the sites based on how deep they were. And from there, we plotted out a grid of 300 meters, and we both had random sites selected, and as well as targeted sites based on the bathymetry that we knew, if there looked like some good rock piles out there, or other things that, other information that we had to go by. And so what that gave us was a target of 12 sites at each port. So for gear selection, there were 2 different gears that were used, but most of the time it was done by the egg pump. We only needed 1 good day to get a whole port done, and it will sample a larger area. We drove the boat, well, Fish and Wildlife Service drove it backwards, and we drove it forwards off the side. But you drive the big weighted sled on the bottom, and it sucks everything up off the bottom, and then you can see it going into our little stepped filters, and then it gets onto these screens. So for the egg pump, we could have repeat replicates in that same day, but again, the weather has to be nice, and we have that 8 meter max depth, and we definitely can have pump malfunctions, which one year we were plagued with. For the sites that USGS did, we regularly sample using egg mats. And so along with the pump sampling, we also threw out some egg mats to see what we could catch. Again, the advantage of that, you don't have to have quite as good weather. We could have deeper sets, which I don't think we did. I think we just sampled in those same grids. But they can stay out there until the weather clears. But if it's really wavy, which it definitely can be on that shore, we could lose the gear. So that's I'd say the main disadvantage for the egg mats. So for the sampling strategy, I mentioned we had targeted sample sites, as well as some random samples. We used those two different gears. Over in the Eastern Basin, they did have some egg mat sampling that they did in 2021, but for the most part, they also did, they used the egg pumps. In 2021, we started sampling around 10 degrees Celsius, which put us in like mid-November. And then based on the results that we found there, we waited until later in 2022 so that we did more sampling in December, when we felt it would be more likely to get whitefish eggs if they were there. And we continued sampling sort of as long as we could, which usually ended about the week before Christmas. We did 3 replicates at each site. And then we tried to visit, the goal was to visit each port 3 times in the sampling season. And that was sometimes difficult, and especially since we wanted to make sure it was cold water at the time. So for the Central Basin, 2 of the crews, USGS out of Ann Arbor and the U.S. Fish and Wildlife Service out of Trenton, split those ports up. We originally wanted to do 3 ports both years, but we found out quickly that between the lake and the sampling, that just was not going to work out. So each year we chose 2 ports out of the list that was there, but Lorain and Fairport were sampled both years. And then the Eastern Basin crew, they sampled these different reefs and sort of known or suspected spawning areas, but they sampled different sites each year. So for sample processing, you can see here the screens that were used, but they just went through a screen and then we would pick through it. So this is stuff from the Eastern Basin. And then over here is what was on a screen from the Central Basin. Right there, that is a Lake Whitefish egg. And any Lake Whitefish eggs that we captured were brought back to the USGS for, which this might be on another slide. Nope, it's not. Okay, any eggs that were captured were brought back to the USGS to try and hatch out. For the habitat, Fish and Wildlife Service went out to those 6 ports in the summer, and they used the side scan sonar, and you can see their path here. And they went through to each of those site grids that we had and scanned with the side scan sonar, as well as having 16 camera drops in each grid for ground truthing. We use a CMEX classification system and took to classify what we found. And then we also took things like water depth, temperature, dissolved oxygen. And those things were also collected during sampling as well. So for results, in 2021, we captured 42 eggs. And in 2022, we had 70 eggs, whitefish eggs specifically. And they were always caught at the Lorain port and the Fairport ports. We also caught lake eggs at Fairport, at Barcelona and Shorehaven, as well as Woodlawn Bar over in the Eastern Basin. And we should note that lake trout eggs, as well as pre-embryos, the first in Lake Erie, were captured at Shorehaven. And our Fairport area is recently has had detailed mapping and sampling by Ohio DNR to identify some walleye spawning in the Central Basin. So it's not just us going out there. There's really multiple species that are using the similar habitats. And we're all learning from each other and sort of where to have targeted efforts for the future. So if we zoom in on where we capture the eggs, here's a up close of our Lorain sites and where they're at in relation to the shoreline. And so for Lorraine, we really had sort of the eggs were captured kind of all along the areas that we identified. And if we compare that to Fairport, Fairport really had these areas that were west of the harbor there. That's where we caught the most eggs for both years. Now, when we look at the water depth as well as the temperature for when we captured the eggs, we captured eggs at from 3.6 to 8 meters depth. So pretty much most of the depths that we were able to sample, we did capture eggs. That's less surprising. But for the temperature, we really didn't catch anything until it hit that 6-degree mark. And we captured eggs from 1.7 to 5.5. So for 2022, again, I mentioned that we delayed when we started sampling, and that was based on what we found in 21, so that we, didn't quite go out there and waste our efforts when whitefish weren't going to be out there. And if we look at a breakdown of the amount of sampling that was done, which are the bars, in relation to the temperatures when the sampling occurred, which is what these little whisker plots are. We have the mean temperature is the X, and then the full range is the whiskers. We can see that we had some sites that weren't really sampled that often below that 6-degree mark, which could be contributing to why we didn't catch any whitefish. And those sites like over here for Lorraine and for Fairport that had both those high sampling efforts, as well as making sure that it was, at those colder temperatures, that those are where we captured our whitefish. And so that could have something to do with why we didn't catch eggs at all the sites that we sampled. Results for the substrate are up here with each bar indicating that a separate 300-meter grid. The stars at the top are where we capture at least one lake whitefish egg. And you can see places like Kanya and Ashtabula, they had a lot of bedrock and those large gravel mixes. And Fairport has a lot of this white in the bars, and that's indicative of our sand. And our Cleveland, Lorraine, and Huron had sort of a mix of all the classification groups. If we zoom in to the places where we actually did capture eggs, for Lorraine, again, I mentioned that there was a mixture, and for a lot of the sites where we captured eggs, it was these bedrock and gravel mixes, except for one site that had a lot of sand, and we have some examples of what those looked like from the drop cameras. And for Fairport, we can see we had a lot of sand, or we captured the eggs in a lot of the sand areas around Fairport. So for all of the eggs that came back to USGS after they were collected in the field, were inspected, and anything that was fungused or looked dead did not go into the hatchery. So even though we caught all those eggs each year, for 2021, we only had 8 eggs that were able to be hatched. And for 2022, we had 13 eggs that were able to be hatched. And those have been sent off to Wendy Stott, and she has been able to run the genetics on those. But we're still awaiting analysis of that part. So for conclusions, this is the first contemporary record of whitefish spawning in the central basin of Lake Erie. We detected eggs at 2 ports, and we detected those both years, Lorraine and Fairport. And Fairport, we caught them at the same individual sites in both years. So that's interesting, and for the future, a place that, we would definitely want to put more effort in those direct areas. We caught them at a variety of depths. Again, we were limited by the pump function. So they are likely out there deeper as well, or could be. And again, the biggest thing was that we didn't get any eggs until it hit at least 6 degrees or lower. They were found on a variety of substrates, both the sand and the gravel. And this is really, I want to say, where they were found, not necessarily where they were spawned. sand and those small gravel that's going to make it easy for the eggs to move around, as well as that those are some of the more effective substrates for the egg pump to sample. But those eggs were still in the area. So maybe they weren't put down right where we caught them, but that's giving us a good starting place for future studies to, narrow down where whitefish are spawning in these areas. And again, we had lake trout spawning that's been documented, or spawning aggregations were found in Fairport, Ohio, as well as out at those reefs in New York. We're at multiple groups and species, using these reefs that we're, everyone's trying to figure out where they're at. And we caught eggs at both those targeted as well as random sites. And for future work, because of how we set up the study, we really wanted to be able to do some occupancy modeling. Jason Fisher is heading that up, and that's still in development. And as I mentioned, we just got our fish to Wendy for the genotyping. And with that, that was all I had for today. I just want to give a huge thanks to the field crews that went out and would go at a moment's notice when it looked like the winds would let us out of some port. And I have my email address if anyone has any other questions.
Okay, thanks so much, Robin. Great talk. And I don't know, Corbin looked like he was having some camera issues earlier, and it's fine if you want to keep your camera off, Corbin. It doesn't really matter. You don't have the bandwidth for it. So yeah, let's open it up to questions. Really two nice complimentary talks. Corbin describing to you what they have just started in terms of describing in a really novel way what that reef habitat might look like under the ice. And really great to see that they had such a good ice year to be able to document that. And then Robin and her team's work documenting for the first time Lake Whitefish spawning in the Central Basin. So if you have questions, you can drop them in the chat, or ideally you can raise your hand and just throw your camera on and ask directly. I'll start off with Robin. So we've got like Cisco restoration or Cisco reintroduction happening in the New York waters of Lake Erie, this year. And those historical maps indicate, of course, there was spawning habitat in the Eastern Basin. So do you think the lack of, and then if we think about whitefish as a surrogate for Cisco, do you think the lack of detection of Lake Whitefish spawning eggs in the Eastern Basin was more to do with sort of the temperature? I don't remember specifically that. Like, do you think if the sampling had occurred when it was colder, 6 degrees and colder, you all might have detected it there? Or just
Yeah,
curious about that.
yeah, I think so. Some of the sampling that was done by the Nature Conservancy and the Lower Great Lakes Office, they were only able to get to the different reefs. Again, they only went to each one of them 1 year, and they weren't necessarily able to get out there as many times because some of those sites were, more offshore than what we were restricting ourselves to in the Central Basin. So I definitely think that there is whitefish spawning out there. That's my opinion based on our larval that we found. We found yolk sac larvae from sampling around Dunkirk. So it's happening somewhere. And, hopefully future sampling will be able to, figure out where, kind of narrow that down, both for whitefish, but also for the Cisco restoration efforts.
Yeah. And the fact that lake trout were using that is another good sign too, in the Eastern Basin. So yeah.
Yeah, they
Yeah,
caught
Roger.
lake trout at a bunch of the sites.
Yeah. Yeah. Yeah, Roger.
Hey everyone, good talks. I guess I had a couple questions, but one was just this validation of spawning by whitefish, just people using nets or even sonar, just and maybe with your cameras, just to see if you can document actual spawning in some of these areas, particularly Niagara Reef. I was wondering about, but the others. So I'll stop with that one. I do have another one after that too.
Yeah, so we I'll say my research group isn't doing that, but I believe that there are people out of the Sandusky office that are looking for the whitefish and any spawning cisco that can be found on the Western Basin reefs as well as in some other areas. So, but that isn't what we do, but they are doing that.
While we were deploying our monitoring stations, there was some gill netting occurring on Niagara, and they did catch some adult whitefish. It seemed to be in spawning condition.
If there's no one else, I'll just follow up with a second question, which is particularly Niagara Reef is a known angling hotspot. So do you have communications to keep the anglers away from your gear, or are you worried about them snagging? I mean, there are a lot of people that jig bottom
I am
lure.
in fact worried about that. So this has been kind of a stressful winter. I was glad we got ice, but then I was also worried about ice crushing our stuff and or displacing it. But we met with the Habitat Task Group before we put these things out, and just my initial plan was to talk to really do like kind of a multi-criteria decision analysis, a really structured way to decide where to put these, to try to avoid those conflicts. But in talking to folks, they were pretty supportive of putting them on Niagara. But so hopefully nobody drops an anchor on them or something, and hopefully we're not creating any frustration with people getting snagged or anything like that.
Thanks, Roger. And Corbin, do you have like video like look like from your really cool animations that you deployed those from the boat, which makes sense rather than diving that time of year, or I can't, or maybe even the depths are beyond ease of diving. But um are you certain, like, how did you secure it so that it didn't land sort of wonky or something like that? And maybe sorry for such a simple, silly question, but do you have like video evidence that it's sitting the right way to collect data and the right sort of dimensions that you wanted to collect the data?
So they have some lead ballasts on the feet, so they
Okay.
in theory should should land bottom down, but we'll check them when we pull them with a camera and just make sure that everything looks like it should.
Okay.
But in theory they should be weighted in a way that they land correctly. And
They're
two,
designed for that, designed that way. Okay. Yeah.
yeah, and also they were tethered until they hit the bottom, and then we released a slip line, so they shouldn't have flipped. But
Yeah.
we will verify that they're oriented correctly when we pull them.
Hey, Scott, welcome.
Thanks, Bo. Good stuff. And this is interesting in relation to work that's being done here in Green Bay, in Wisconsin waters of Green Bay, with whitefish egg detection on our reefs and in the Fox River by either the Fish and Wildlife Service or EW Stevens Point. So it's really going to be interesting to compare those results. But I have a quick question for Robin. And I may have missed this, but it's two parts. So the first part, if you answer it a certain way, it may negate the second part. But were these the results you showed a compilation of the egg mats and the suction, or was it just for the suction sampling, the numbers that you had shown? I might have missed that somewhere.
Yeah, I believe all the figures were all just the egg pumps.
Okay.
But I think the total numbers that I reported were or they were for both.
Okay.
So which would really only affect the Lorraine site. But we did catch eggs using the egg mats.
Okay. I wondered if in terms of like the distribution, because I was thinking that egg predation from gobies, for example, on egg mats could have obviously could affect the numbers that you would see, and if that could affect if those were distributed differently, the sampling was different for the different sites, may have some effect to just theoretically thinking that if you just use egg mats or if they were predominantly used at one site, that egg predation by gobies in particular could be an issue. It could affect those results. But if it was mostly you're showing was egg suction sampling, then it might not be as big an issue. But yeah.
Yeah, we did not have as many egg mats. So at our sites, I don't believe we had egg mats at every site that we did an egg pump for. I think we picked a subset of those just because the sampling really did take every minute of daylight, and you know needed everything to be as calm as possible for the egg pump to work well.
Uh,
So the mats, I would have to look at the sites if they're the same that got eggs using the egg mat and the egg pump. But I don't know that answer offhand.
okay. Yeah, I'm just struggling with some issues too with different sampling we've done on our tributaries with different gears too. Or not struggling, but curious about the effects. So this is all good stuff. Really cool. Thanks.
Mhm.
Robin, you're kind of leading do I remember right that you're leading a team that's looking into different ways to sample like eggs of whitefish specifically? Like there's egg traps, egg mats, the pumping, sort of the advantages and disadvantages of all of those. And you're all working on maybe a review paper for that? If you want to give a plug too,
Yes.
or
Yep, we are. And that's on the list. But it's
okay.
yeah, it's specific to carrigines, and it covers, we're gonna have all the different methods that have been used for whitefish, as well as if there's a few extra ones, and we'll have pictures and dimensions. We're really just trying to make it be sort of a um, like almost manual kind of thing, a real like source of information based on what your habits. So we're gonna have it broken down by um, which gears work well in different habitats, kind of caveats you need to think about. And then Ellen Marsden is leading a complementary one looking at lake trout. And so the two are kind of like sister papers to kind of go over the two things and why. And we're going to share an online supplemental that has like all the pictures and the real details of each gear.
Yeah. Thanks for that. And then, so Scott, that might be something that would be helpful for some of the issues you're coming up with, and others maybe on the webinar. So
Yeah, absolutely. I mean, there's like work being done, but like I think 3 or 4 different kinds of gear being used to sample eggs right now in Green Bay and in the tributaries. So that will be very important information to have, very relevant for us. Thanks.
other questions? Corbin, I may have missed it, but are the data you're collecting sort of first of a kind in the Great Lakes, or has this been done in other systems that sort of inspired it, or can you give us some context for how unique these data will be?
So there was a paper that came out relatively recently that um, seems to be similar. I'll have to look more closely at that paper because I'm not sure if they left the moorings out over winter. Um, but it's not uncommon to collect these sort of data throughout other parts of the year. But I'm not sure they've been collected during the winter. We weren't able to really find much when we were developing our proposal. Um, and I don't think generally folks are targeting reefs for these. So I
Yeah,
don't.
definitely super relevant for whitefish and for the shallow water ciscos. So
Yeah, I was excited last time during the last webinar because a lot of this stuff folks seem to be discussing and thought, well, we're collecting some of this. I should tell people.
exactly, yeah. Any other questions or comments? Okay. Well, if not, we hope to see you again on May 2nd. Uh, we're going to actually keep the focus right on Lake Erie and be hearing about some habitat suitability models from Caitlin King, as Renee mentioned, and then some population viability models by Laura Lee, to help contribute to um, the Cisco reintroduction, experimental reintroduction happening in the New York waters of Lake Erie. So thanks again to our speakers today, to Robin and Corbin, for stepping up, filling a void that we had. Great presentations, seeing some positive feedback online. And everybody have a great weekend.